Hey everyone, I'm Dr. Snexer, and this week we're going to be talking about reading scientific papers and summarizing articles. I want you to pause this and think for a second about scientific papers, about reading the primary literature. When you think about reading scientific articles, how does that make you feel? What emotions are you feeling as you're thinking about reading these papers? Are you excited? Are you scared? Are you anxious? Are you overwhelmed? What are your feelings associated with reading scientific articles? What is your comfort level when it comes to reading scientific articles? Do you get a paper from a professor and think, yeah, I got this, no problem? Or you start sweating at the mere thought of having to read yet another scientific paper with all of that jargon and all those crazy words? When you read articles, are you assessing them, really taking them in for the science that they're representing, or are you just trying to make it out alive? Think about this for a minute. Now that you've thought about it, let's talk about how important it is to be able to read and evaluate the primary literature. As we are becoming research scientists, we realize how important it is to read the work of other scientists within our field and within other fields, those that are related to us and maybe those that we don't know a lot about. We should be able to read and evaluate all types of scientific papers, even if we're not necessarily familiar with that species, with that technique, with the topic that the paper is about. We should be able to read the articles and evaluate all that is within those papers. In order to do this well, we need to fully understand the research process, and so understanding how research questions are asked and how hypotheses are proposed, understanding methodologies, those that are common, or just general patterns to how we utilize different methods or techniques within a field, how to collect data, understanding the different variables in both observational and experimental contexts, being able to analyze and interpret data, to have a basic understanding of statistical analyses, and how we ourselves would develop a project related to whatever it is we're reading about. Those are all really important aspects of being able to understand the primary literature. So the more familiar you are with every part of this process of science, the easier it will be to read these scientific articles that, let's be honest, when we look at them, sometimes are a little intimidating. So today I'm going to go through the basic parts of a scientific article, the basic patterns that they follow, and then give you some hints on how I read scientific articles and how I think you can become better at reading the primary literature. Now, when I say scientific literature or primary literature, what I'm really talking about are peer-reviewed journal articles. Now, scientists report their results in a variety of different ways. They could do this at scientific conventions called conferences. They could write up reports. They could write a book or a chapter in a book. Of course, if you're working on a master's or a PhD, you often write a thesis or a dissertation. But what we mainly look at as scientists is the primary literature, which are peer-reviewed journals that have gone through a rigorous process of review by other scientists within the field. Sometimes we call peer-reviewed journal articles primary literature because it's the primary source. It's the scientists who actually did the work that are reporting on that work, that are explaining the results, and so not just a reiteration of it from a new source or a different scientist who's reinterpreting the results of the original scientist. So when we're looking at primary literature. We're talking about the person, the scientist that actually did the work. We also sometimes call these journal articles refereed or scholarly articles because, as I said, these are reviewed by other scientists. These are not just written by the original scientist and put out into the world, but other scientists will evaluate each article and determine if it meets the quality standards that the journal has set forth. So is it a good experiment? Are they asking an interesting question? Did they control for the proper variables? Are the conclusions being made in that paper actually related to the data that were collected? Or are we jumping to conclusions in a sense? All of these things are taken into consideration in that peer review process. Now, I should point out that not everything in peer-reviewed journals are necessarily peer-reviewed or primary literature. You can pick up Science or Nature or Animal Behavior or Behavioral Ecology, any of these journals, and while the bulk of the material in there are going to be primary peer-reviewed research articles, there are also editorials. There are letters to the editor from other scientists. There may be comments on, very exciting things that are currently happening within the field. And often there are review articles, which are useful. We will definitely talk more about review articles in another video. But they are not peer-reviewed, or they are peer-reviewed, but they are not primary. So other scientists will look at them, but reviews are exactly that, a review of many different scientific experiments that have happened and are sort of being brought together in an interesting way to look at one perspective or one idea. So it wouldn't be considered primary literature. Very useful. Love research articles, review articles, but they are not primary literature. So that's what we're focusing on today, though. The primary literature, research articles that are reporting the results of a scientific experiment that has been done. Now, of course, these peer-reviewed primary literature articles go through the entire peer review process. So scientists do their work, they get their results, they write up those results, and then they send them off to a specific journal. So a specific scientific journal that is appropriate for that work. Medical journal, an ecology journal, a cell biology journal, a cognition and learning journal. So usually scientists are sending their work to specific journals that are based within their field. The editor receives the information or receives the manuscript that the scientists have written up and then sends that out to other scientists within that field. So within the field, there will be many scientists that work on similar ideas, and editors will identify those scientists and send them the information. Those scientists will review the material for free. This is not a paid service. Scientists do this as part of their scholarly work. They review articles of other scientists, and they evaluate them to determine if the science is sound. Did they do the proper experiment? Are they actually answering the question that they say they're answering? And then they'll provide feedback to the scientists and the editor, and they'll send that back. The editor will review that. Usually what happens is the editor then sends those comments back to the scientist, who has to make some adjustments, whether it's to the experiment itself or just the way that it's written. But only then will it actually become accepted into that journal. Now, depending on the journal, there's a good number of manuscripts that get rejected at this point, that don't get published because they need a bit more work. And the scientists will have to go back and try again. This is like writing a draft for your professor, and they just cover the whole thing in red pen. But that usually ends up creating a better scientist and a better scientific experiment and a better paper in the end. So this is a tried and true process that's been going on for a long time And this is how we get those scholarly articles published in those journals. So let's talk about actually reading the scientific articles. So how do we actually read an article now that we have it? How do we get through it? How are we taking the information within this article and actually understanding what it's about and assessing it for ourselves? Assessing to see if the science in here is telling us something new and exciting now that it's been published. So in order to do this, I have some tips. I have some tips for you as you get started and as you go through dozens and dozens and dozens of articles across your scientific career. Probably hundreds and thousands at some point. Okay, so you get an article. Maybe you download it, maybe your professor gave it to you, you found it on the web, a friend sent it to you, you've got your article, and now you're ready to read this and understand it. What do you do first? Before you dive in, pay attention to what the article is really about, and the title is really going to tell you that first. So look at the article title first. Take a look at the authors. It's always important to have some sort of context, who the authors are. Maybe you've read their work before. Maybe you're familiar with them. Pay attention to the authors, what institutions they're from. And, of course, be able to identify what journal the article is published within. And really just all the information that's available to you within the citation for that paper. So every paper can be cited, right? We can give a reference for every article that we're reading. You've probably written up a bibliography or a reference page in your lifetime. There are certain rules about formatting these things. I've included here an APA formatted citation for the paper we will be looking at. APA is the American Psychological Association, which is closest to the style that is used within my field of animal behavior and behavioral ecology. Each journal in this field has specific requirements on how to write up these citations for articles, but basically they all include the same information: the author's last names, the year it was published, the title, the journal, the volume, and the page numbers that the article can be found on. And you can usually find that on the very first page of the article itself. So here's the article that I cited on the previous slide. And like I said, this is just the very first page that you would get if you picked up the paper version of this article or if you downloaded a PDF off the internet. You see here we've got our title. This paper is about the social partner preferences of male and female fighting fish, Betta splendens. You can see that it is published in the Journal of Behavioral Processes. This was published in the year 2006. It was in volume number 72, and it spanned pages 38 to 41. So you've got all the journal information right here on the first page. And no matter what journal you're looking at, you can generally find this information right at the beginning, or maybe it's at the bottom of the first page, but it's right there on the front page of the article. You can also see the authors. Oh, look here, J.L. Snexer, who at the time was at the Department of Biology at St. Joseph's University, along with S.P. McRobert, and their co-author, E.D. Klottfelter, who is still at Amherst College in Amherst, Massachusetts. So look, you can see a whole lot of information right here. We haven't even gotten into the article yet. We've already got all of our citation articles, and we're starting to get an idea of what this is about, right? This is going to be about the behavior of Betta splendens. You probably know these as bettas. And looking at social behaviors of males and females. So the title is a good place to start. But once you've got the title down, what else do you need to go through? Well, most scientific papers follow the same basic structure. If you've ever written up a lab report in any science lab that you were in, you probably followed the same basic structure. Sometimes this is called IMRAD because IMRD. But generally speaking, all scientific articles have an abstract, an introduction, methods, results, discussion, and then finally the references that they use throughout, that they're citing throughout. Sometimes this might be called a bibliography, but generally these are your citations or your references that you cited. It's called IMRAD because the bulk of the structure of the paper is, of course, in the introduction, methods, results, and discussion. So let's talk quickly about each of these sections. You're probably familiar with them, but just to remind you, let's talk about what's in each one of these. First is your abstract. This appears usually the very first paragraph right at the top of the page. You could see that on the front page of the Beta Splendens paper that I showed you. The very first thing underneath the authors and the title was the abstract. What's the abstract? It's a summary. It's giving you like the super quick version of what that paper is about. The most important findings, maybe the research question that was being asked. Just really, if you only had 5 minutes to read a paper, you'd hit up the abstract first. It's 150 to 250 words. Each journal actually will have a word limit for their abstract and that the authors must abide by. So it's going to be short and sweet and to the point. Right? Every paper starts with an abstract. A lot of research conference presentations, poster presentations, and oral presentations generally have an abstract too. So this is just a summary of what the work is really about. When we get into the bulk of the paper, we start with the introduction. The introduction is a literature review. It puts that scientific study into context. So why is this being done? What is the historical relevance of this? What other studies have been done that are related to this specific project? And then for this specific project, what is the research question that is being asked? And what hypotheses and predictions are being made? Now, not all scientists are just going to come out and say, and we hypothesized that, and we predicted that. Sometimes you have to be a little bit of a sleuth to get in there and figure out exactly what the research question is, exactly what hypothesis is being tested. A lot of scientists put this sort of near the end of the introduction, maybe in the last paragraph or so. But it should be within there somewhere, along with this larger literature review that is putting the paper into context. The second section in most papers is the methods. Now, I should pause for a second and say occasionally the methods will be included at the very end, or even as a supplemental material now that a lot of journals are strictly online. I personally think methods are important, and so including them as supplemental information might cause you to miss it. So make sure you look. If you've downloaded an article and you don't see the methods, they may have been part of the supplemental information, or they might be at the end near the references. But for the most part, you'll find the methods right after the introduction, and the methods are exactly what they sound like. They're just describing the design of the research that was done. What variables were tested, who participated in this, some sort of description of that. If it's humans, we'll understand what group of humans were looked at. If it's animals or plants or some other species, there's usually some sort of description of that species, where they're found, maybe some life history traits. Basic measures of the populations that were studied, and then of course the actual procedure that was done. So step by step we did these things. You'll also often find that scientists will reference other research articles when it comes to their protocols or procedures, especially if they're using something that's standard. So if they're using a protocol, especially in like the molecular side of biology, they'll just cite sort of the standard for that protocol in the paper, with the assumption that most scientists will know what that is if they're in the field. Sometimes you have to go and look up those papers if you really want to understand the methods, but there will be at least some basic description of what the methods are within this methods section. The next section are the results, and the results are where you actually find the data that were collected during this study that you're reading about. So I wouldn't necessarily say they're findings in a conclusion way, but they're findings in a numerical way. So they're quantitative or qualitative findings, usually some statistics. Most peer-reviewed articles are going to include some sort of statistics, whether they're just descriptives like mean, standard errors, sample sizes, or actual statistical analyses like t-tests, ANOVAs, correlations, regressions, or other fancy statistical analyses that are comparing groups. This is where you're going to find them within the results. I think more importantly, within the results, you're going to find the graphs and the tables. And in a minute, I'll tell you, these are very important. The graphs and the tables are going to display the important information that was collected during this study. So graphs and tables within this results section, very, very important. Now, following the results, we have our discussion, and the discussion usually starts out with a quick summary of what the results said. So just a paragraph that said, our main findings were X, Y, and Z. Then it's followed with an interpretation of what that means. So within the context of the organism that we're studying or the research question that we're trying to answer, what conclusions can we actually draw from the data that we collected? And then some scientists go into, strengths and weaknesses, how they could have done things better. Some don't. Right? Some don't necessarily analyze the project, but instead look forward to the next step, sort of what we can ask now that we know this bit of information. So what are the future experiments that will be done? You can find all of these in the discussion. A good discussion is also going to relate back to the introduction. Now I say a good discussion because not all discussions necessarily do that, but well-written discussions circle back to what was introduced at the beginning of the paper, bringing up that research question again and talking about the hypotheses that were proposed and whether or not the data supported that hypothesis or refuted that hypothesis. So generally we look for those things in the discussion. You may have been familiar with all that before. This is a general structure of how scientists write papers. Now, this is how a paper is written. This is how a paper is published. This is how you will look at a paper. It will have these basic sections. But how do we actually read papers? Think about this. How do you read a paper? You sit down, get all cozy, get your mug of coffee, your hot cocoa, and you're ready to just dive into this scientific paper. Do you read the abstract and then go through to the intro and then read the methods and then the results and then the discussion and then glance at the references? Maybe you do. I don't. That is not how I read scientific papers. So I, along with most scientists, have to read a lot of scientific papers. We need to keep up in our field to know what other scientists are doing. We want to learn more about fields that are related to what we're doing. And we often do not have the time to read the entire paper from top to bottom. Now, if we love a paper and it's very relevant and important to us, we will. We will go back and we will do that. But sort of the first go at a paper usually isn't from the first word to the last word. So, how do we do it? Well, here's my tips for you. 1, I do start at the abstract. That's the truth. I do start. I read the title, I look at who the authors are, and then I read the abstract. Why do I start at the abstract? It gives me a good summary of what the paper is about, and then honestly, it tells me if I should waste any more of my time with this paper. If I read the title and I think it's going to be relevant to the project that I'm working on, and then I read the abstract and I find, this isn't really what I thought this paper was about, I don't bother reading the rest of it. If it's not relevant or not interesting or just isn't going to serve a function that I need at the moment, the abstract's going to tell me that right away. And so I'll read that abstract. And it's only like 200 words. So how long does that take me to read the abstract? Not very long. Gives me a good idea of the paper, tells me if it's worth my time or not. Sometimes I don't move on from that. Right? Get rid of the paper. Get rid of it. We don't need it anymore. But if it's good. Now let's say I read the abstract and I think, this one's relevant. This one's important. I want to spend some more time on this. Where do I go next? I go to the results. I go right to those results. I know you're like, but Dr. Snexer, what about the intro? How do I know what this is about? Well, the abstract told you a little bit about what it's about. So I want to go to the bulk of it. I want to go to the important information of this paper in the graphs. The graphs are where it's at, right? The graphs and tables, depending on the paper that we're looking at. Graphs, tables, figures. Scientists put all the important information in those figures. You know how they say a picture's worth 1,000 words? Yeah, it's true. Show me those figures, because those are all of the methods and the introduction and the research question all together in the findings, right? In the findings of what these scientists actually studied. So by looking at the results, by looking at the figures, I can get an idea of the methods, right? Of what they did based on the variables that they are presenting within their figures. I'll show you an example of this in a second, but looking at the X and Y axes of a graph tells you exactly what variables were tested. Reading that figure caption at the bottom tells you the groups that are being compared to each other. And so I don't have to read through the entire method section unless I need a little bit more info, right? I want to know what the sample size is. I want to know what time of day these animals were being tested. I want to know where the people were that were being surveyed. So if I want a little more info, I'll hop from the results to the methods just so that I can get a better idea of exactly what's going on. But I don't necessarily read through all of the methods first. I start with those results, I see what's important, and then I skim through the methods to get a general idea of the experimental procedures if I can't understand those from the figures. Then maybe I'll read the introduction. I usually focus on the last paragraph or two of the introduction because that's where I'm going to find the hypothesis. That's where I'm going to find why the scientists did this study and what they're trying to figure out. Yes, it's great, especially early in your career, to read the whole introduction because it will give you an idea of the historical context of why the study is being done. But if you're very familiar with the topic, if you've already researched this organism for a while, you probably know most of the information that's in that introduction. And so after you've read a few papers, you'll spend less and less time in the introduction. Early on, when you're starting a new project, you probably spend a bit more time. So when I start studying a new organism, or if I start just some new aspect, some new study, I will spend more time in the introduction. But if it's something I'm very familiar with, then I really only look at the last paragraph or two to understand what the scientists were trying to do. After that, I'll hop to the discussion. I know, all over the place, right? But the discussion's great, especially the beginning of the discussion, because it will summarize what's in those figures. So those figures, the very important findings that came out of the study are shown in those figures, and they are usually summarized within the first paragraph or maybe the first two paragraphs of the discussion. And so it's always good to look there, but I wait to go to the discussion until after I've looked at those figures and tried to understand exactly what was being done within the study. Does that sound crazy, all of that? Maybe. Maybe it is. But this is how a lot of scientists get through papers pretty quickly. In an average week, if I'm working on a big project, maybe I'm writing up a manuscript, or maybe I'm trying to find papers to present in class, I'll read a dozen or so scientific papers. Will I read them from the first word to the last word? No, I can't. I can't do that. But I will use this method to get through the papers and understand them. If it is a paper that is really important to what I'm doing, of course I'll spend more time with it. But this is a good way to get a very basic understanding of what's going on so that you can really assess this paper and determine, did these scientists do a good job? Did they really do what they were supposed to be doing? Did they really test the hypothesis they were trying to test? You don't have to read the whole paper to know that. You can jump around a bit and figure it out. So let me use an example to try and get you more comfortable with reading papers like this. So going back to our paper on the social partner preferences of male and female fighting fish, Betta splendens. If you're not familiar, I put up this picture because this is what they look like. They're adorable. Betta are well known within the pet trade because males have these elaborate traits. They have long fins. They usually have beautiful blues and reds and purples, sometimes white in that like iridescent, beautiful shimmery color. So they're very elaborate, very ornamented. And they're well known in the pet field because they are generally solitary. They're called Siamese fighting fish because if two males are put together, they will fight each other. They start out with what are called opercular flares. So they flare out their gill flaps. And then it can even lead to biting. And if they're incredibly aggressive, it can even lead to death. And so when getting a betta as a pet, you often just keep one male all by himself. I mean, really, that's what you're supposed to do. Just one male in a cup all by himself, away from other male bettas because they will fight with each other. Females, this is a picture of a female Betta splendens, are very drab in comparison. You can see they're usually like whitish, maybe some grayish, purplish color to them. They do not have those long, elaborate fins. And they don't have the same sort of aggressive nature that we see in those males. But my colleagues and I noticed long ago that nobody was really looking at sort of the sociality of these fish. So we know that you're not supposed to put bettas together when you buy them at the pet store. But do bettas really prefer to be by themselves, or is there some sort of social aspect to their behavior? Because a lot of fish, even closely related fish to the betta, are very social. They prefer to hang out in groups, which are called shoals. And so we wanted to explore if there were any sort of social partner preferences of male and female Betta splendens, hence our title. So we were looking to explore that research question, and you'll be able to read through this abstract and find our general questions of what's going on, right? So that's why I said, if I'm interested in the title, this is about social behavior in a fish that's actually known for being asocial. Okay, so let's see what this abstract is all about. It says social interactions have been studied in the context of dominance hierarchies, mate choice and communication. What about social preferences? It's been largely overlooked. Okay, anytime you see a scientist say something's been largely overlooked, you know that's what this paper is going to be about. So here we studied male and female betta fish in different contexts, with a male, with a single female, and with a group of females in a dichotomous choice test. Oh, look, there's some methods there to better understand basic social interactions in this largely non-social species. And then we get some results. There's a highly territorial, the highly territorial betta preferred associating with conspecifics, that means fish of the same species, in nearly every configuration we tested, except for these specific ones. And in most tests, they preferred to actually be with the larger group of females when given the choice. So this is very interesting because while such behavior might not suggest true shoaling behavior, it does demonstrate a subtle degree of sociality. Okay, so look at this abstract. I got a good idea about what this is about. There were some fish, males and females. They put them into different situations. They were put into this method of a dichotomous choice test, so they were given 2 choices. And then these scientists looked to see what choices did they make. The results indicated that they're maybe not as non-social as we thought. Okay, I'm hooked. This sounds thrilling, doesn't it? Thrilling. You know I'm the author on it, so I find it interesting. This was actually the first paper I published as a master's student long ago, which is why I chose it for y'all, because it's pretty basic. It's pretty straightforward. It's not too complicated, and the results are pretty obvious once you look at them. So what do I mean, look at them? The graphs. Show me the graphs. I told you, after the abstract, I read the methods. Show me the graphs. This is figure 1 right here. And in figure 1, when you look at it, you can see a whole bunch of information right away. And I won't go through all this because I'd like you to look at this paper yourselves and try to understand a bit about these figures. But just looking at figure 1A and figure 1B, we can start to get an understanding of what the methods were and how that relates to our hypothesis, right? Our research question was understanding if these fish were social or not. And so we tested A, which were the females, and I would write females right on here. And then we tested B, which were the males, and I would write males right on here. And we tested them in 5 different testing situations, and those are all laid out for you right here within the figure legend, right? Within the caption of the figure. And so you can go through and you can say, okay, test number 1 was a single female versus an empty chamber. Test number 2 was a group of 3 females versus an empty chamber. Figure number 3 was a single male versus an empty chamber. Okay, so already you've got an idea of what's going on. Here we've got females, and they're given a choice. Now you can go back to the methods if you're not familiar with what a dichotomous choice test is. It's basically a fish tank where you have 1 choice on the left and 1 choice on the right. 1 female fish versus nobody. 3 female fish versus nobody. A single male fish versus nobody. Right? And where do the fish want to hang out? That's what mean association time means. So this is your average time in seconds that the female fish are spending with either the female fish or on the empty side of the tank. Plus or minus standard error, which are these little bars here. And look, there's some stars. And if you go into the paper itself, I know it cuts off here at the bottom, but if you go into the paper, you'll see those stars indicate some sort of statistical significance where there's a difference. There's a choice. The math tells us these females like to hang out with single females rather than being by themselves. These females like to hang out with a group of females rather than being by themselves. Oh, but let's look at this one. What's this? This is interesting. When given the choice between an empty container, an empty chamber, and a male, what did the females choose? Where did they spend more time? Okay, so I'm going to let you look at both of these graphs and think about that. But the point is, look at these 2 graphs. They're chock full of information. You didn't really spend a ton of time in the methods. You didn't read through all the results. You didn't have to because a picture is worth 1,000 words. Right here in this picture, reading the figure legend, the caption, you can understand the basic methodology and the basic results and conclusions that can be made from this paper. So this is where I always start. If I want to know about a scientific experiment, I go to figure 1, figure 2, figure 3, table 1, table 2. That's going to give me the basic information that I need. And that's what I encourage you to do. Whether it's this paper on Beta Splendens that I wrote many, many moons ago, or another scientific paper, by looking at the abstract to get a basic idea of what the paper is about, and then moving to the figures to really get an idea of what is being measured, what is being tested, you can get a basic idea of what the paper is really all about. Now, how do I get there? How do I get good at this? How do I read 12 papers in a week like Dr. Snexer does? Practice, practice, practice. I wish there was an easier answer. There isn't. You just have to practice. The more papers you read, the easier it is to read papers. The more papers you read, the easier it is to read papers. Just like any other activity you do in life, the more you do it, the better you get at it. It's like a sport. Reading these research papers, if you practice, you're going to get great at it. The more papers you can read, the easier it will be for you to read papers. Now, specifically in this course, we're going to do 8 article summaries. You're going to actually start with this one that we've been talking about today. So you're going to start with this Beta Splendens paper. You're going to summarize it for me. You're going to tell me what it's about. And I kind of know what it's about already. So I'm looking to see if you can figure out what this paper is about. And you should do this independently on your own. You can ask your classmates if you need a little bit of help. You can ask me questions. But you really should try to work this through on your own to see if you can get the general idea of what this paper is about. Following this, we will actually have 7 other article summaries due. So they're going to be due on Tuesdays before we meet in class. And those 7 other articles are going to be articles of your choosing. So you will get to decide what you want to read about, which articles you want to summarize, and they should be related in some way to some of the ideas you have about your research proposal. So whatever research question you're going to come up with, start finding papers that are related to that. Say you want to study non-social species in a social context. Well, I bet there's a ton of papers about that. Say you want to read about ladybugs. You're going to be able to find a ton of paper about that. Maybe you want to study ferret behavior. Maybe you want to study how people with snakes as pets do in other aspects of their life. I have no idea what you're going to want to research, but you're going to look for papers that are somehow related to that research question, and you're going to summarize those over the next few months. And I'm really excited because you'll be teaching me a whole lot of new science because I won't know the answers to all these. So I'm relying on you to summarize these papers. So when I'm grading your summaries, I'm going to be looking to see if you've done this, if you've gone through, you've looked at the figures, you've figured out a bit about the research question that's being asked, a little bit about the methods that they use to collect that, and just a general idea of what the paper is actually all about. And I provide you with a framework for doing this. So you'll see this PDF or this Word document posted up on D2L. And I want you to use this as your framework for every article summary that you do, starting with the Snexer et al. 2006 paper. For each article that you read and you summarize, and I recommend you do this for all articles in the future, I still use this when I summarize all my articles so that I don't forget what they're about and I don't have to reread them later. I do a quick little article summary. I write down the full citation. I always use the APA format. If you're not familiar with that, I can give you some resources so that you know how to cite it properly. But I write down the full title and all the information about it. I usually jot down then the hypothesis that I think that is being tested after I read the abstract, but then I'll jump over to the main findings, right? Because that's the graphs. That's how I read papers. So I want to look at the graphs and I want to summarize the main ideas, not everything they found in the paper, just the main ideas of the paper. Sometimes I'll even draw a little bit of the graph in there so that I can get an idea of what they were looking at. I'll then go back from those results. I'll be able to get an idea of the basic methodology, what animal was being looked at, or was it people or plants, I suppose. How did they test it? Like, what are the basic methods? Again, just a sentence or two. A dichotomous choice test, a PCR reaction, an observational study in which animals were looked at 20 times a week, whatever it is. I'll write a very brief summary of the methods. At that point, I should have a pretty good idea what hypotheses were being tested. I will double check that by looking at the introduction. And then I'll glance at the discussion, usually the beginning of the discussion, to find out what was the importance of the study? Why was this study done? And relate that back to the hypothesis. Then I always make little notes if there are other notes that I personally think about this. Maybe I think, wow, this is a really great study. They did a great job controlling for X, Y, and Z. Or maybe I think, I need to look and see if there's anything more recent than this, right? Like, oh, this 2006 paper about betas, has anybody else looked at it since then? So maybe I'll make that as a note. Or maybe I'll think like, oh, I didn't really like this paper, or their methods weren't great. And I'll write down those notes, or I'll relate it to other studies that I've read. So this is just a way to organize my thoughts, and I think this is a great way for you to start organizing your thoughts. And then just collect all of your summaries. I keep them together in electronic format, and then I can pull from those as I'm looking for those ideas in the future. So maybe you'll start out thinking that you're going to write your research proposal about, pet owners who own snakes, and then you decide, actually I'm going to look at pet owners with turtles. And so you won't necessarily use that snake paper that you read, but maybe 2 or 3 years from now you will. So it's always important to keep these summaries so that you have this sort of reference library at your fingertips whenever you need it. So I hope this is helpful as we get started, as you're summarizing first this article, and then as the weeks go on, other articles that you found for yourself. We will search for articles, and I will help you to do that. And I've actually posted a few extra videos this week from the Canisius College Library. Our librarian, Matt Koshin, who's wonderful, has made us a couple videos about looking up scientific articles and trying to use the search engines from our library to find what we're looking for. So I'm going to talk more about that in class on Tuesday. We'll go through some examples ourselves, but if you want to get a jump start on that, please watch the other videos by Canisius College Librarian Matt Koshin. It's wonderful. Of course, you should be reading this paper, this Nexer et al. 2006 paper that I went through in this video, and complete your article summary, which can be uploaded to D2L in the Dropbox folder as a PDF or a .doc file. If you have questions on this, just let me know. It's due before class on Tuesday. So it's due before class starts on Tuesday. You can just upload it, and we'll actually talk about it in class on Tuesday. Also, don't forget, there is a discussion board due this week that is just an opportunity for you to share some of your research ideas and to comment on others' posts about their research ideas. So please make sure you do that this week. As always, if you have any questions, please don't hesitate to let me know.